One of the most primitive cells that originated on Earth were the bacterial cells. Life and cells have evolved so much since then. Now the cells look so much more different in their complexity of structure and function. But these primitive cells, the prokaryotic cells, were the basis of life. Hello everyone, welcome to Manocha Academy. And in today's video, we are going to talk about those primitive cells, that is the bacterial cells, and look at their structure and function. A bacterial cell is a typical prokaryotic cell that we see around us. What is a prokaryotic cell? As we all know, a cell which does not have a true nucleus but has a genetic material in the form of a nucleoid, they are known as prokaryotic cells. Let me break this down. What is a true nucleus? A true nucleus is a structure that contains four parts. A proper nuclear membrane demarcating the nucleus from the rest of the protoplasm, nuclear reticulum, nucleoplasm and nucleolus. In case of a bacterial cell, we see that these structures are absent. So what do they have instead? They have cytoplasm in which a naked DNA without any protein is floating. This structure is known as the nucleoid, and all cells that have this kind of genetic material, they are known as prokaryotic cells. The word pro means primitive and karyotic comes from the word karyon meaning nucleus. So these cells have a primitive type of nucleus. Bacterial cells can be classified into two types on the basis of their reaction to Gram stain. Christian Gram developed a method of staining bacterial cells with the help of a 2 and 3 step method. In the first step, bacterial cells are treated with gentian violet or crystal violet. Usually, all the bacterial cells take up this color and turn blue. The bacterial colonies in your medium on a petri dish will appear bluish in color. After this, the same cells are treated with 0.5% iodine solution, following which the cells are now washed in water and then in acetone. Now we get two types of reactions from bacterial cells after these three steps. Some of the bacterial cells completely lose the blue color once they are washed in water and acetone, and some of the bacterial cells retain that blue color even after being washed by water and acetone. Those cells which retain the color, they are known as gram-positive bacteria, and those cells which lose the blue color are known as gram-negative bacteria. Now, what is the reason behind this differential reaction towards the same chemical? The reason lies in the bacterial cell itself. Some of the bacterial cells have a very high lipid content in their cell wall and some don't. Now this cell wall is responsible for taking up the blue color. But those cell walls which have very high lipid content, the lipid dissolves when the cells are washed in acetone because lipid is soluble in organic solvents. As a result of this, some of these bacteria lose the blue color when the cell wall with high lipid content dissolves, and those with cell wall with low lipid content retain the blue color even after washing. A bacterial cell, like any other cell, is protected by an envelope. The cell envelope in case of a bacterial cell consists of three parts: the outermost glycocalyx, the cell wall and the innermost cell membrane. The glycocalyx is a layer which consists of two parts, the inner S layer and the outer layer of mucilage. The inner S layer is made up of proteins and several other chemicals. This layer is responsible for protecting the bacteria from the harmful action of enzymes of hosts or show immunogenic reactions, that is, evoke the immune system to react against the bacteria, or it helps the bacteria to attach to the host cell or to the host body, and it also helps the bacteria to get its own characteristic feature. The outer layer, which is made up of mucilage, is basically a carbohydrate derivative and is known as the capsule. This gives bacteria a sticky nature which helps the bacteria to attach to the hosts. Along with the S layer, the capsule is also responsible for similar functions that the S layer shows. So what are the functions of the glycocalyx? First and foremost, it prevents desiccation. That is, it prevents the bacterial cell from drying up because mucilage itself is a slimy, watery fluid. Second, it helps the bacterial cell to be protected from the harmful enzymes of the host. So as a host, my body would like to kill a bacterial cell that has entered into my body. But if the bacterial cell wants to be protected from my enzymatic actions, the glycocalyx will help. Third, the bacterial cell, with the help of its glycocalyx, is capable of showing immunogenicity. What is the meaning of that? It means that our immune system reacts to the bacterial capsule, that is to the bacterial glycocalyx, and identifies it as a foreign substance. So fourth point is, it also gives bacteria its virulence. Meaning that if my body is capable of identifying the chemicals that are present in the glycocalyx as a foreign chemical, then my body will try to kill the bacteria because it's a foreign cell. But if my body is incapable of identifying the chemicals present in the glycocalyx as foreign, the bacteria is protected from the harmful action of my immune system. Such a bacteria will be able to hide in my body and therefore cause a disease. We call such bacteria virulent bacteria. In such bacteria, the glycocalyx protects the bacteria from being identified by our immune system, so the bacteria can stay in my body and keep on showing harmful reactions. 5. The most important point, if you are consuming any kind of drugs, which is medicines, to kill a bacterial cell, the capsule might protect the bacterial cell from the harmful chemical of the drugs. The next layer that is present inside the glycocalyx and outside the cell membrane is the cell wall. Now plant cells also have their cell wall, but such a cell wall is chemically extremely different from the cell wall of bacterial cells. In case of a bacterial cell, the gram-positive bacteria contains a single-layered cell wall, whereas the gram-negative bacteria contains double-layered cell wall. The outer layer in gram-negative bacteria is rich in lipid, and that is the reason why when we wash it with acetone after gram staining, the lipid washes away along with the stain. The inner layer of the cell wall of a gram-negative bacteria and the single cell wall of gram-positive bacteria are mainly made up of peptidoglycans. So what is a peptidoglycan? The peptidoglycan, as the name suggests, has two components, peptido or peptide and glycan or carbohydrate. Now what is a peptidoglycan made up of? It is made up of repeated units of N-acetyl glucosamine alternating with N-acetyl muramic acid. The N-acetyl glucosamine, which is the main component of peptidoglycan, is basically a carbohydrate derivative which contains an amide group, that is an NH2 group. And there are peptide linkages in between. So this is basically a combination of protein and carbohydrate. In case of both gram-positive and gram-negative bacteria, peptidoglycan, muramic acid makes up the major constitution of the cell wall. The innermost layer and the third layer in case of a bacterial cell is the cell membrane. And this cell membrane has the similar structure as we see in eukaryotic cells. We will go into the details of the structure of this cell membrane in another lesson. But to let you know about it, the cell membrane is made up of lipid and proteins. It has proteins embedded in the lipid bilayer, floating outside the lipid bilayer, and the most important thing about the cell membrane in a bacteria is that this cell membrane is highly metabolically active and several reactions are taking place inside the cell membrane. Inside the cell membrane, like every cell, bacterial cell also has a jelly-like fluid which is known as cytoplasm. The cytoplasm in a bacterial cell is devoid of membrane-bound cell organelles, especially double membrane-bound cell organelles. But non-membranous cell organelles like ribosomes are present. They also have other cell organelles like vacuoles which store different substances. One of the most important structures present in a bacterial cell is its mesosome. Mesosomes are inner foldings of cell membranes which form a structure like this. Mesosomes can be of two types, septal mesosomes and lateral mesosomes. The septal mesosomes have been shown to form cell membrane after cell division. So it is believed that these mesosomes are responsible for development of cell membrane or membrane biogenesis. Lateral mesosomes are connected to the nucleoid, that is the genetic material, and the plasma membrane. So it is believed that the lateral mesosomes are responsible for division of the nucleoid. One of the most important functions that mesosomes perform is respiratory reaction. And majority of the respiratory reaction takes place in the mesosomes, because of which mesosomes are thought to be the precursors of mitochondria. In case of prokaryotic cells, they do not have mitochondria. But the function of mitochondria and mesosomes look very similar. Hence, mesosomes are also known as incipient mitochondria. Having said that, respiratory enzymes are present all over the cell membrane. So it is not only localized in the mesosomes. The next cell organelle that we are going to talk about are the ribosomes. Now, ribosomes are those cell organelles which are present both in prokaryotic and in eukaryotic cells. They are membraneless and are made up of RNA, that is ribonucleic acid, and proteins. In case of a prokaryotic cell, the ribosomes are slightly different from those in eukaryotic cells. In both the types of cells, the ribosomes are made up of two subunits, a larger subunit and a smaller subunit. These two subunits come together during the process of protein synthesis. In case of prokaryotic cells, the larger subunit has a sedimentation coefficient of 50S and the smaller subunit has a sedimentation coefficient of 30S. But when you join them together, their sedimentation coefficient is 70S. So in case of prokaryotes, the sedimentation coefficient of the entire ribosome is 70S, whereas its individual units are 50 and 30 for the larger and the smaller subunit respectively. Please do not get confused here. The 70S value of the entire ribosomal structure is not coming by adding up 50S and 30S. Then it should have been 80S. 70S is the value at which the entire ribosome sediments when you are carrying out ultracentrifugation. Whereas if you take the two units separately, they sediment at different rates. So 70S, 50S, 30S, they are separate rates and we do not add up 50 and 30 to get 70. The ribosomes are the protein synthesizing machinery. So in case of both prokaryotic and eukaryotic cells, we see that a message comes from the DNA that this is the protein that needs to be synthesized. That message goes to the ribosome. The ribosome reads the message and prepares the protein. Now this was said in a very simple sentence, but this function is extremely complex. So when DNA wants to synthesize a protein, the information about protein synthesis, which means which are the amino acids that need to be joined and in what sequence, that information is present in the DNA. From the DNA, the information has to go to the ribosome. But the entire DNA cannot go to different ribosomes. So what happens is, that region from where the protein has to be made, that region is converted into a small RNA, that is ribonucleic acid. This RNA carries the message from DNA to the ribosome, and that is why it is known as mRNA. So the DNA forms a small mRNA which has the code or the information about protein synthesis. This messenger RNA goes to the ribosome. The ribosome attaches to the messenger RNA, reads it and joins amino acids one after the other to form the protein. During this time, one messenger RNA can have one ribosome attached to it, or in prokaryotic cells we see that one messenger RNA has many ribosomes attached to it to make multiple copies of the same protein. Now imagine it looks something like this, where you have an mRNA and many ribosomes attached to it like a string of beads. This is known as polyribosome. It is found only in prokaryotic cells. In eukaryotic cells, polyribosomes are not really found. Certain bacteria are photosynthetic, but they do not have double membrane bound or membrane bound structures. That is why chloroplasts are lacking in them. They do have membrane bound sacs which are known as chromatophores that contain bacterial photosynthetic pigments. In some bacteria, the membrane that covers the chromatophores are typical. That is, they have that protein lipid structure as we see in cell membrane. But in some, they are atypical, that is, they are not proteinaceous or made up of lipids. The pigments that are present inside these membranes are usually bacteriochlorophyll, bacteriophyophytin and carotenoids. In photosynthetic bacteria, chromatophores acts as a site of synthesis of food. Let us come to the most important part of any cell, its genetic content. As we have already discussed, bacterial cells are prokaryotic, so they do not have a true nucleus which is separated from the rest of the cytoplasm by a nuclear membrane. So what do they have? In the cytoplasm, there is a slightly more denser region which contains a naked DNA. This naked DNA is called naked because it is not coiled around any protein, or they do not have a scaffolding of protein around them like we see in case of eukaryotic cells. In our cells, DNA always remains in association with proteins and forms a structure called chromatin or chromosomes. But in case of prokaryotic cells, this is not seen. The DNA is naked and the single DNA is equivalent to one chromosome that we see in eukaryotic cells. So you have 46 chromosomes in all the cells of your body except egg or sperm, and each of these chromosomes contain one DNA molecule. In the bacterial cell, imagine that they have one chromosome, which means one DNA molecule, but they do not have a chromosome structure. They only have the DNA without any protein. In case of bacteria, the DNA is usually circular and double-stranded. This double-stranded DNA, along with the region of cytoplasm that it is in, is known as the nucleoid, or an incipient nucleus or a primitive type of nucleus. It may remain attached directly to the cell membrane, or it may remain attached to the cell membrane through the mesosomes. Apart from this DNA that is present in the nucleoid, bacterial cells are also known to have extra chromosomal DNA. That is, DNA present outside what is supposed to be a chromosome, in this case the nucleoid. These are known as plasmids. The plasmids are much smaller than the main DNA. They are double-stranded and they are self-replicating. So if the plasmid wants to make a copy of itself, it can do it on its own. It does not have to depend on the DNA of the cell to do so. Now, what is the utility of a plasmid for the bacteria? These plasmids, they have some special genes which might not be absolutely essential for the bacteria to survive, to reproduce or to get nourishment. They provide the bacteria with some added advantage. For example, you must have heard about this, that I am developing antibiotic resistance, meaning I am taking antibiotics so much so that now the same antibiotic is not being able to kill the bacteria that is inside me. Now this requires a gene for the bacteria. So a bacteria, let's say, is resistant to tetracycline, which is an antibiotic. The gene for that resistance, known as the tetracycline resistance gene, is present in this plasmid. So most of the antibiotic resistance genes of bacteria, they are present in this structure, which is the plasmid. Apart from that, the plasmid also contains genes for fertility factor, which means that these genes will determine if the bacterial cell will be able to carry out sexual reproduction with another bacterial cell and how. Plasmids are also useful for us. We can use these plasmids in a process called genetic engineering, about which you will learn in your later classes. In certain group of bacteria like purple bacteria, cyanobacteria, etc., there are special vacuoles which help in storage of gas. These vacuoles are known as gas vacuoles. They are surrounded by a non-unit membrane, that is a membrane which is not made up of lipid and protein, and these gas vacuoles are responsible for orienting the bacterial cells, providing it with buoyancy so that it can float in different depths in the water depending on what time of the day it is. That allows the bacterial cells to be able to reach the region of water where sunlight is available and therefore photosynthesize. Apart from this, the gas vacuoles also protect the bacterial cells from harmful radiations. Coming to the different structures that project from a bacterial cell. So we have three types of structures that project from the bacterial cell: flagella, cilia or pili and fimbri. Let's talk about flagella first. Usually these are long whip-like structures which may occur singly or in tufts in different places of a bacterial cell. Now a flagella in case of a bacteria is equivalent to one proteinaceous microtubule. It is in case of eukaryotic cells, most of these kind of flagella we find that there are tufts of microtubules, but this one is equivalent to a single microtubular structure. This flagella has three regions: the basal body, which remains embedded in the cell envelope; the hook, which is a curved proteinaceous structure and is the thickest part of the flagella; and the flagellar filament, which is made up of proteins flagellin. The basal body, as you can see, is made up of several ring-like proteins that are arranged inside the cell envelope and anchors the entire flagella to the cell envelope. The hook is curved. It is also proteinaceous and attaches the basal body to the actual filament. The filament is made up of flagellin proteins. The flagellin proteins are globular, that is, they are spherical in structure, but they are arranged one after the other to form filaments which remain spirally coiled to form a structure like this. The entire flagellar filament is made up of this kind of arrangement of flagellin protein. The flagella, with the help of its movement, which takes place in somewhat this kind of a manner, helps the bacteria to move from one place to the other. The two appendages, pili and fimbri, have been confused as the same structure for a very long time. Pili are actually appendages or projections that come out from the cell of bacteria which help in sexual reproduction. So during sexual reproduction, when two bacterial cells come close to each other, they have to form a connection between the two, and that is known as a conjugation tube. This is a structure through which the genetic material of one bacterial cell travels to another bacterial cell to get mixed or shuffled. So the pili is responsible for forming that conjugation tube during sexual reproduction in bacteria. Fimbri, on the other hand, are cellular projections that have been found to help the bacteria to attach or adhere to the hosts. The bacterial cells also adhere to each other with the help of these fimbri, but they do not help in forming conjugation tube. Alright, so that was all about a prokaryotic cell or a bacterial cell for this video. Now let's see if you can find this answer. Can you tell me the name of a prokaryotic cell which is devoid of a cell wall? Do let me know your answer in the comments below. Looking forward to reading your answers. I really hope that you are enjoying these videos. Do hit the like button and share it with your friends if you do. If you haven't subscribed to our YouTube channel yet, hit the subscribe button right now and click on the notification bell. Do check out the full courses on our website, Android app, Manocha Academy. Links are given below. I really hope that we will stay connected and we will keep learning together.